So I managed to confuse the lovely IT guys here because they were expecting Peter Wildy to give that lecture. So they couldn't quite work out what I was doing when I was trying to get my slides sorted. But thank you very much for the introduction. Before I get going, I would like to thank the people who've helped me stand in front of you today with receiving this prize. When you're a scientist and you get an award like this, it's important to recognise that it's not just you that's managed to get you to where you are, that there's a lot of work that goes on with support from other people. And in particular, I'd like to thank the University of East Anglia, the Microbiology Society, and the wider microbiology community for all their encouragement and support. And without it, I genuinely wouldn't be here talking to you today. Now, the clue to my talk is in the title, and I'm just going to start off a little bit by telling you about me and how I got involved in microbiology. So I'm going to take you back in history to Uganda. So I moved to Uganda in 1968, more than 50 years ago, even before I was born, as I tell my daughters, because they have no idea how old I am. And it was because my father was a scientist at the time working with soil. And Uganda was a post-colonial country in the middle of East Africa, and he was stationed there to work with a lot of other scientists from across the globe to understand and work with the Ugandans to try and make their soil as productive as it could be. So when you fly into Uganda at that time, you flew to Entebbe, which was the local airport just outside Kampala, and then we would go cross-country through the Zika forest to the research station, which was in a place called Serere, which was in the middle of Uganda, and it was quite a remote area, and the only people that were in it really were the Ugandans working on the research station and the scientists who had come from across the globe. So it was a really interesting time, and I had an amazing childhood. It was even though we were sort of quite remote, there was a lot of freedom. We spent time with lots of the other children roaming around. And I was very mindful that there were lots of rules that you had to follow at the time. So poor Huskisson always thinks I'm a bit of a wuss because I'm terrified of snakes. But that came from our childhood because we had some really close encounters with poisonous snakes at the time. But the other thing that we were also told as the children was that whenever we went out, we had to make sure that we kept shoes on because they were creatures that could get through your skin and work their way through your veins and lodge in your organs. If you left your toys outside overnight and it rained, those toys automatically had to go in the bin. We always had to sleep with mosquito nets, and also we were never allowed to drink water unless we knew where it had come from. And in our case, it was water that had been kept in the kitchen in metal containers that we knew had been boiled and purified. So that was a really fantastic time. And I remember thinking, how come there are all these rules? How come we have to be so careful of things that you just couldn't see? Because it meant that you had to have lots of imagination. And I was very mindful too that we could see quite often the result of tropical people and animals that had tropical diseases that had left them really profoundly affected going forward. And it was this question that sort of started in my childhood that stayed with me through my education. So we returned to the UK after the arrival of this fella, which some of you in the audience might recognise, but he's one of the worst dictators that Africa ever had, and his name was Idi Amin. And what he wanted to do was to get rid of everybody in Uganda that wasn't Ugandan, basically, and it was due to him that the whole middle-class tier system that there was in Uganda had to leave, so the Ugandan Asian community were all left with nothing except the clothes that they were standing up in at the time. And also lots of the other governments, they took out their scientists because they recognized that there was real danger here. So the Danish, the Dutch, the American, and the Canadian, they actually came into Surrey and they flew out their scientists who were working there with their families. The British government was a bit more laissez-faire about it. They just told us to make sure our car was kept ready, to have blankets and pillows in the boot, and that they would contact us by radio if things just started to escalate and take off. And that day came, so we left. We went across the Kenyan border, and my mum and us, we all flew home, but my dad was left there to finish his contract, which was with the Overseas Development Agency. So a few schools later, because you hear about forces children, well, I was a scientist's child, so we toured around a lot, even when we got back to the UK, because my father was always on quite short-term contracts. And I think I worked it out that I had been in about 7 schools before I left school and went to university. So I went to university in St Andrews, which was I left school with a batch of hires when I was 16 and went straight to university, where I studied microbiology and biochemistry and really enjoyed it, and took a year out after I finished because I wasn't the best student. I wasn't really sure what I wanted to do, but I managed to earn some money and did a wee bit of traveling. And it was during that time that I actually met another group of scientists who had done masters and were actually working in overseas development. And they just gave me that push or that nudge to think about going back to university to do other degrees, which I did. I didn't go very far, but I went to the University of Dundee to work in the laboratory of David Boxer. And it was there that I met Frank Sargent and Tracy Palmer, who are friends and colleagues who I still know to this day. And it was also while I was at the University of Dundee, where I was working on looking at how E. coli can grow aerobically and anaerobically. So I was working on the hydrogenase enzyme at that time, that I met this fella. And we eventually got together about 2 months before he left to go to Houston, Texas. So it was at that stage that I had a decision I had to make. I'd just started a postdoc in David Box's lab, and I had to make the decision, do I stay and do my postdoc in Dundee, or do I take a risk and move to America to be with my then partner? So decision made. It was a couple of months after that that I moved to America and we got married. And you can see here that we got married with the people who were witnesses were people whose lab Richard was working in at the time. And again, a lovely multicultural wedding service we had there. And very shortly after that, I had my daughter. So everybody says to me, oh gosh, did you get married because you were pregnant? And I was like, no, I got married because I needed my visa to get into America. So. Yep, but then, we had a fantastic time. I loved my time in Houston. It was such a privilege to be there. I really enjoyed every minute of it. But we had to make a decision, and that decision was whether we decided to stay in America and take that route, or whether we decided to come back to the UK. And we had lots of ties in the UK. And eventually, Richard, my husband, got another job. So we'd spent 2 years in America before we moved back to the UK. And I had another child, and in the end, I think I had a career break that lasted for about 4 years. So I'd been out of the workplace for 4 years, having completed a PhD with probably about 3 or 4 months of postdoc to my name. And at that time, I thought, my goodness, this is going to be interesting. I'm going to have to find something to do because I really couldn't see science opening up in front of me. I thought I'd burnt that bridge. But we eventually moved to Norwich, which is a fine city, and we moved there in 1998 because my husband got a job, and it was very exciting because this was a 5-year appointment, so it meant that we had permanent roots for somewhere for 5 years. And Norwich is a lovely place to be. It's a very modest city, it's a very pretty city, and we're famous for lots of things, including our cathedrals and our castle. Delia Smith, who's a fantastic cook there. We have mustard, Coleman's mustard. We have a fantastic football team who hopefully is going to get promoted. And then also Alan Partridge. So Alan Partridge, if you don't know, is a spoof character. So he is epitomised as someone who works in local radio, and he's entirely crass. And Norwich people, they don't know whether to really love him or really hate him, because in some ways he puts Norwich on the map, but sometimes he puts them on the map in a way that they don't really like. So he's a character. But it was shortly after that Stefan Bornemann, and I have to give him credit for this, took a gamble, and he employed me on a short-term, 6 months, part-time contract as a research assistant, because his permanent research assistant had gone on long-term sick leave. And I have to be forever grateful for that. And it was, for both of us, I think, a really good decision, because that job soon turned into an 80% RA, and I was there for 8 years in the end, and things lasted really well. And during that time, I began to establish myself, again, as a scientist and a microbiologist. So I started to publish papers, which are always a big tick if you think you want to put yourself over as being a good scientist. I really established a network. There were lots of people at the John Innes Centre who I have still got contact with today. And it was during that time that I became intrigued by AMR. And that was due to a joint project that I had with Stefan Bornemann and Professor Mervyn Bibb, who wasn't in my department, but was in a department that was in the same building. And I would like to say this opportunity that this is Tracy Palmer, who again had moved to Norwich, and her PhD student at the time, Nicola Stanley Wall, and I would like to thank Nicola for nominating me for this award here today. So it was during my time at the John Innes Centre that I began to communicate science. John Innes Centre has a real history, a track record of communicating the science that they did. And I started to get very, very interested in the whole reason behind why communicating science is a good idea, getting members of the public in to explain to them what you're doing and to find out what they think about the work that's going on. And especially because the John Innes Centre is part of the NRP and it's on the edge of the city. So people had to make a real effort to come and see you there. Now, 2006 was an interesting year for me, and I think it was one of those trigger years that changed history, in my opinion, for myself. And it was because the British Science Festival came to Norwich. The British Science Festival is the biggest science festival that the UK has. It goes on a tour every single year, and if you have an opportunity to take part in it or go, please do, because it's fantastic. But they asked me to be the biological sciences representative, and that meant that I had to work with staff and colleagues at the John Innes Centre to design and deliver events that were part of the festival. And it was during this time that I realised I really wasn't feeling confident enough to do events myself, to go up and talk to people like I'm doing now. But I was actually really good at persuading other people to go and do talks to members of the public. And you can see here that that's what had happened. So the photograph in the middle there is my husband, who was very easily persuadable, because I had lots of ways to persuade him. And then also Tony Maxwell and Mark Butner too, who were all doing work around AMR. And the very first proper event that I did was working on AMR, and that was way back in 2006. Now, because Richard and I were both at that event, we had 2 children at the time, so our kids always had to come along with us. And you can see here my daughter Ellie in the green T-shirt with the BA Festival of Science on, handing out the food at the end of that event that took place in the middle of the city. Now, what's really interesting is that we have done lots of events together, and our children have gone to loads of that event. So we've actually practiced what we've preached about taking our daughters and trying to get them involved in STEM. And we've had a 0% success rate. So neither of our daughters are doing STEM subjects at all. They're both doing social sciences and psychology. So I don't know what that says about us at all, really, or me in particular. But eventually, when I was working at the John Innes Centre, I was only ever 80%. So I always said yes to opportunities. And I had 2 great opportunities. So the first was to be work at UEA doing some teaching for the School of Biological Sciences. And also I picked up a role working with the Open University as an associate lecturer, talking about molecular biology and again, the cell biology as well. And what that did was it introduced me to this wealth of people who had come into higher education through a completely different route. These people had families, they had full-time jobs, but they were committed to getting an education. And I was absolutely inspired by the students that signed up for this. And it also opened my eyes and made me realize that actually going into higher education is not a straightforward route for a lot of people. And it's something that stuck with me, and I really try hard to opening doors to higher education to people that want to go, but perhaps hadn't thought of it. I'm not saying higher education is for everybody, because I genuinely don't believe that, but I think everybody, if they want to be able to go, should have the opportunities to be able to take part in it. So 2007, after the BA Festival of Science, made me think, maybe there's other jobs out for me. And I applied for a job at the University of East Anglia, which I got, so I had the confidence to apply, and I was really shocked to get this job. But it meant leaving the John Innes Centre on one side of the NRP, crossing the River Yare, and ending up at the University of East Anglia, where I still am today. So UEA is a university in a fine city, and it's a fine university. It's a beautiful campus that had been gifted to the city. The city had gifted it to the university because they believed that higher education, having somewhere where there was higher education, was really important if the city was to grow and be successful. And I've got here a picture of the rabbits, because we're absolutely famous for the UEA rabbits as well. And it's springtime now, and these little baby rabbits are starting to pop up all over the campus. And this one was so sort of bold that you were able to go right up to it and take a picture of it. So it's a gorgeous campus to visit, and we'd love to see you if you decide you want to come and visit UEA at any time. And my job was in the Norwich Medical School, which was a brand new medical school set up in 2002, and it was expanding. And this was during the expansion time when the government recognised that it needed more doctors. So way back in 2002, and lo and behold, it still sees it needs more doctors. But I got a job there, and my job was a teaching fellow. So I knew that my role was to teach the students microbiology, to inspire them to recognise that microbes are something that they should be studying. But it was a decision I didn't take lightly, because it meant that I had to leave my research career behind, because they were very specific that in this job you can't do both. It was one or the other. So I thought, well, I'll take a gamble, because you just never know what might happen and things can change. So I moved over there and suddenly thought, my gosh, I'm a very good microbiologist. I've got all the degrees to show that I can do this. I've got my BSc, my master's, my PhD. I've got my papers, but I have nothing that says that I can do education or teach very well. So I decided to do the master's in higher educational practice. So I came to university and did that whilst I was a lecturer, so got another degree, and felt that that gave me the confidence and actually a real knowledge and understanding about the field of education. It's not just as simple as turning up and giving a talk like this in front of a group of people and expecting them to be knowledgeable at the end of it. Learning's much more complicated than that. And it was during that time that my science communication really took off, and I was lucky enough to be teaching on a module called Science Communication in the School of Biological Sciences with my colleague Kay Yeoman, who we still work together, and we recognised that there was no book out there on science communication for scientists. There was lots for social scientists who study how scientists do communication, but none that were there giving some tips and tricks and ways forward, lessons learned for how you can start doing science communication well. So that's a book there, and if you're interested in it yourself, it's in all local good bookshops, and not so good ones too. So my academic career, I can sum it up in 4 different phrases, and that is antimicrobial resistance. It's something that I'm still very passionate about and teach. Widening participation to higher education. I still do lots of science communication. And another thing that I have been involved in all the way through is equality and diversity. So my background and the experiences that I had as a scientist establishing myself and getting a job in higher education meant that this was something that was particularly resonated with me, is making sure that all females have the same access to success as their male colleagues. And I'm currently the person who's putting together and just literally finishing the final sentences of the Athena Swan Silver Award that's going in at the end of April. I'm going to fast forward 10 years, and I managed to get a chair in 2016, so I became a professor, and my professor title was Professor of Microbiology, Education and Engagement. And I have to say that I have complete respect and gratitude to the UEA for giving me opportunities to really succeed and to begin to develop as an academic. And just recently, last year, I was seconded out of the medical school, and I'm currently working as the academic director for innovation at UEA. So this is across the university, working with lots of colleagues from the humanities, the social sciences, the sciences as well as the medical sciences, at looking at how we support and build up connections with our outside community and other key stakeholders. And the other thing that I'm doing too is supporting our impact case studies across these sectors going forward. And that is such a privilege, working with people who are doing fascinating research in areas that I just think are amazing and so interesting to find out about. But my whole career wouldn't have happened without the Microbiology Society, and I say that from the heart. One of the first things that I would like to tell you about is my role as the editor-in-chief for Microbiology Today, which is the best magazine of any society that there is out there. Absolutely. And this was a fantastic role. And the reason that I took it on was thanks to the characters that you see in this picture, which are Matt Hutchings and Paul Huskisson, who had been editors previously, and they gave me the confidence to apply for this editorship. And this is my favourite ever edition, sorry boys, but I think this one is still the one that is up there as one of the best that there was today, which was having the whole idea that microbes had superhero qualities, and I still have a real soft spot for that particular edition. And this gave me an opportunity to mix and meet microbiologists who I would never have come into contact with, who are working on such a diverse, amazing projects. And the thing I'm most proud of, I think, in my role there was the opportunities I gave for sort of early career scientists, PhD students and postdocs to write articles for that magazine. And we had lots of excellent articles from our younger career scientists. The other thing was that I joined the education division, as it was then, which soon morphed into the communications committee. And it was at that time that I met Jo Verrin, who was the chair of that committee at the time. And Jo has been, and I want to give her a real a name check here, really, because she has been a mentor. She doesn't know she's my mentor, but she's absolutely been a mentor for me and has been one of the people who I just admire hugely. And I think people understand that when you saw the Twitter feed about Jo's session yesterday. And it was during that time that we had an annual conference in Manchester, which I think was in about 2012-13, but we did our first AMR education session there, which was very, very popular. So joining the Microbiology Society gave me a real opportunity to really begin to embed education and to see that it was something that had links to education and microbiology. It could bring them together. Also Hilary. So Hilary chaired the Equality and Diversity Working Group in the Microbiology Society at that time, and I was part of that working group, and we, through that, developed the Equality and Diversity Policy Statement. And there's still a legacy with that, because wandering around the conference today, and there's lots of little children wandering around too, and I think that's because of the work that that group did. So I think that was a group that I'm very, very proud of. And it also had outreach money, so we could apply for outreach money, and my first ever outreach grant, which I got in 2009, was a Microbiology Society outreach grant. So there's money available, people, if you want to get involved and do this kind of thing yourself. But the sort of one of the passions that I have at the moment, and what really drives me on, is the whole idea about infection. So growing up in Uganda, I was very, very mindful of infection. And I want to take us back to from where we are today to 1900s USA. The Americans are fantastic at gathering statistics, and what they had done is they looked at statistics for morality and actually morbidity, but here we have morality. So these are the certificates of the way that people died in 1900 America, so roughly about 120 years ago. And just to circle the diseases that were actually caused by infectious diseases. So you can see that 4 out of the top 10 causes of death were by infectious diseases at that time. Now, that sort of feels like it's history, that is something in the past. And I understand that. And that's roughly because, well, one of the main reasons is because of this fella here, Alexander Fleming, who is, I think, my absolute favorite scientist in the world ever. And partly that's because he's a microbiologist, like me, that he's a Scottish scientist working in England, like me, that he was also, when I see his bench like this, I think my bench at the John Innes used to look like this all the time as well. But he was an amazing scientist, and he also has a local connection. So he had a holiday home in Tuddenham, which is just over the Norfolk border in Suffolk, but it's on the A11, and that's where he used to go every summer for 6 weeks. Remember those times when you could go on holiday for 6 weeks during the summer and take time out? And he came back to the lab and found his plate, his seminal plate, which had been contaminated. Now, if that had been me in Stefan's lab, I'd have kind of picked it up and put it in the bin and hoped that he hadn't noticed that I'd left my desk so untidy being away for all that time. But not him. He had the foresight to look at it and say, there's something really exciting in here. And that discovery, that sort of sense of looking at things with different eyes is what led to the discovery of antibiotics. And it felt like every single week after that, new antibiotics were found. Microbiologists were going out there and scanning soil samples and looking in the sea for different antibiotics, and they were finding them. And then you had the biochemists who were tweaking the natural antibiotics that were out there and making new compounds that were equally as successful. And you had the chemists who were thinking, well, how can we produce these in vast quantities and do it through factory environments as well? So you had lots of people working on there to have real success stories. And actually, our larder was full of drugs that were effective and working, and we could use them to treat the infectious diseases that beforehand had wiped out populations. And in fact, in 1969, it was a time of great optimism in science. So we'd put men on the moon. It felt like science could change everything. And this is the US Surgeon General William Stewart, who told Congress that we should close the book on infectious diseases. Now, I realize that I've quoted him a wee bit out of context, and it wasn't quite as black and white as that. But nevertheless, it was a genuine feeling that infectious diseases weren't anything that we had to be concerned about at all. But actually, that, I think, was a very first-world optimistic view of everything. And this is a terrible WHO statistics graph. It's really hard, I don't know, to get it to copy properly. But what I just want to show to you is that the graph, with just one arrow on it, is the top 10 causes of death that are caused by infections in first-world countries. But in 2012, if you look at the other side, there's 1, 2, 3, 4, 5, 6, 7 of the main causes of death in third-world countries in present time are still due to infectious diseases. So they're a problem, and they're still a problem in huge amounts of the globe. And let's think about what the kind of infections are that they cause. So Alexander Fleming was working on Staphylococcus. It was his favorite bacteria of the time, and it was a pathogenic bacterium that he knew caused particularly nasty infections and could cause something like septicemia as well. Well, Staphylococcus, they are bacteria that live on our skin. They particularly like mucous membranes, so they live up your nose and in your groin. Most of the time, they're particularly harmless. We all have Staphylococcus on us, but sometimes they can get into the wrong place, and that's when problems happen. So this is a picture of a 2-year-old girl. So if anybody that's had 2-year-olds know that they're infection factories. They have infections all the while. And this wee girl had had a cold, which meant her eczema had flared up. Her nose was running. She'd wiped it into her broken skin and ended up with something called impetigo. Now, left to its own devices, give 6 or 8 weeks, and that infection would clear up and nothing would happen. But actually, she was in nursery, so she'd gone to the doctors to get it looked at, and they'd prescribed amoxicillin. And amoxicillin was a great drug for her. It was an antibiotic, but it worked. And if you looked at a plate which had been taken from a swab, then you saw that Staphylococcus epidermis was destroyed by this particular antibiotic. And that's what she got given. And this is for the most part of you, 60% of the audience have this particular Staphylococcus on your skin. So it's not a problem at all. And you can see here that, in fact, once the antibiotics kicked in, that it cleared up completely. There's no scarring left, and I know that because that was my daughter. And I want to thank her for letting me show that picture of her with her impetigo. But not all children are as lucky as that. And what these are is histopathology slides taken from children who had got Staphylococcus aureus infection. And unfortunately, they hadn't got treated in time, and these children had all died. So it is a really worrying bacterium if it gets into the wrong place. And what is even more worrying is that it's one of these bacteria as well that actually have become well known as a superbug. So we now have methicillin-resistant Staph aureus. For a long time, we could treat it with penicillin, not anymore. So here we have a graph, a snapshot, if you like, taken from 2010, looking at the MRSA isolates in participating countries. So we have got a lot of countries out there that have really high levels of methicillin-resistant superbugs happening. And these aren't the only superbugs or superbug pathogens that we have. We have a good few others. So we have the escape pathogens. But again, this isn't a finite list. These are worrying ones because they're the ones that are associated with hospital-acquired infections, and more and more levels of them are starting to come through that are highly resistant to the antibiotics that we have. But we also have things like TB, we have gonorrhea. So the list of worrying pathogenic bacteria continues to grow, and the antibiotics that they're resistant to continue to grow as well. They were magic bullets, antibiotics, and luckily they still are, but they're not the be-all and end-all that they were. And if we had just listened to Alexander Fleming in 1945, he predicted this. And he said himself that there is the danger that the ignorant man may easily underdose himself, and if he does that, then actually he's going to make bacteria resistant by doing that. And he was right. And he was right enough that what we have now is a report. So Dame Sally Davis was the medical officer for England, and she's a public health doctor and has been very, very good at raising the profile of AMR. When I was working in the John Innes Centre, scientists knew that there was a problem, but that message hadn't gone out past the laboratory door, really. And what she did was she raised awareness of it, and a report came out which was driven by Lord James O'Neill, who was an economist. And I just want to take you to the purple petal of the flower, because you can see here that the next petal down, cancer, at the moment is the biggest killer that we have at the moment. But the purple petal is the killing of deaths caused by antimicrobial resistant infections that is going to happen in 2050 if nothing changes. The little blue strip at the bottom is where we are now, but you can see here in 31 years' time that the biggest killer that we have out there, it's not cancer, it's infectious diseases. It's the 1900s all over again, and I tell my medical students this, and it comes as a real shock to them. It's not something they ever thought about. And the really annoying thing about this report, except for the sort of the bad news that it's bringing us, is the fact that it was commissioned by David Cameron. And I feel so annoyed that I have to give David Cameron credit for anything, but I do have to give him credit for saying that that report should be written. So how quickly does this happen? Well, what I have here is some of my favorite antibiotics. And this is when they were discovered, so you can see them coming up here. And the C is when they went and made it into the clinic. So you can see that we've got lots of them coming into clinic between the 1940s and 60s. We had, like I said to you, a whole batch of them coming up. And then over time, it's been harder to find new ones. But just look at when the resistance to these antibiotics started to become clinically relevant as well. So the resistance starts to come, and in fact, in some cases, there were resistant infections before those antibiotics even made it to the clinic. So this is worrying, and this is the position we're in today if nothing changes. So what we have to do is to increase antibiotic discovery. We need new ones, and also to change behavior, to reduce the way that we're using antibiotics, because anything we can do to reduce the use of antibiotics will push back that time frame. So perhaps not in 2050, perhaps it will be 2060 or 2070 if we can just work together to do this. And I've started working on both those areas, and lots of that has been driven by the Microbiology Society. And one of the projects that I have at the moment is Antibiotics Unearthed, which is a citizen science project. It was a PhD, and in 2014, the Microbiology Society put out a call to do a PhD, which was looking at a project called Antibiotics Unearthed. And in this project, what we did is we got members of the public to give us soil samples, and we did pop-up events where they could bring soil samples to us, and we ran them through the lab. We did lots of testing for them, and during that time we kept contact with the members of the public and just spoke to them about what they knew about AMR and what were their thoughts and feelings about it. And at the end of it, had anything changed because they'd taken part in this project? So it was a really innovative project at the time, and it was a project that I did with Gary Rowley from the School of Biological Sciences at UEA, and also Eleanor Nardi, who's from the School of Education. So completely interdisciplinary. And you can see here my PhD student who's asking me about Antibiotic Unearthed t-shirt on. And also I have to say a big thank you to all the other helpers who came along to the events and really supported us with this project. And again, it was run through the Microbiology Society. So I'm going to summarize all that work, that 3 years' work, in this wee slide here. So we ran 5 pop-up events. We had soil samples collected from 454 members of the public. We picked colonies from soiled plates, so ones that looked vaguely exciting, and we had 929 of those. Colonies that had activity against tester strains that were mock pathogens, we had 165 of them, 15 of which we've looked at through whole genome sequencing because they were particularly good ones. And 3 of them we're analysing further because we can see that they've got stuff in their genome that does look very, very exciting. So during that time, we have done 42 posts for the Facebook page that went along with that PhD. We completed and analysed 32 interviews with members of the public, and we've also got 5 different members of the public to keep a diary for 6 months, our antibiotic diary, and they've returned that to us, and we're currently analysing it. So it's been a really useful project, and we're writing it up at the moment. So if you want to find out the results of it, please do look out for that when it comes through. And I just feel really pleased about it. And we're currently in the process of applying for more money to carry on with this project and to hopefully get a follow-on PhD from it too, because the things that we've looked at are so sort of interesting to us. So we've done, what I do is try and genuinely trying to find a new antibiotic, and I've absolutely hoping that it comes through, because thanks to USS, I need to top up my pension. So I reckon that's going to be about the right time for that. And what I'm also trying to do is to raise awareness of antibiotics. And I do that by giving talks. And you can see a talk that I've given here called Antibiotic Resistance, The Microbes Fight Back. And this is one I did at New Scientist Live down in the EdExcel Centre in London. What else have I done? I've written a book. Here, anybody interested in a book? And that's called The Microbes Fight Back. It's a popular science book. Again, it's available on good bookshops. And in this book, I tell the whole story about antibiotic and antibiotic discovery, and also explain why we're in the position that we're in at the moment. And what I've done here is to do a picture check of my family. So that's my brother who designed the cover. In fact, he designed the cover and did the artwork for both of my books. And my sister, who is kind enough to go through it all and to read it and tell me if it made sense to her, because she's a linguist, she's not a scientist. So if it made sense to her, then I kind of felt I was pitching it right. Science Cafe, oh yes, we do science cafes, so we are quite good at science cafes down in Suffolk and Norfolk. And this is a science cafe we did with an artist who was able to graphically record the talk that we gave. And this is a really neat way, because not only am I talking to you today, but she'd be on the side drawing it. So if you got bored listening to me, you could just watch what she was doing. But again, it was finding out the message that gets across. And these science cafes are still going, and we usually get about 100 people going along to them at any time, and they're really good. The other thing that I work in a medical school, and it sounds a mad thing to say, but lots of the clinicians and the healthcare workers that I know about, they know antibiotic resistance is a problem, but they're not fully aware, unless you're an infection control nurse or a microbiologist clinician who's doing it. But they still need information. So what we did at the University of East Anglia, and I led on this, was to design a MOOC, which was called Using Infection Control to Combat Antimicrobial Resistance. So trying to explain to clinicians, also nurses working in hospital environments, why taking care over infection control is a real way of making a difference when it comes to fighting antimicrobial resistance. And that is because if you can just lower the number of infections that you spread, then the use of antibiotics goes down as well. And that is absolutely something that we need to do. And so far, it's on its 7th run. We've had 2,500 joiners, and that is from across the globe. And all kinds of different people have signed up to this as well, including people who work in care homes. So I'm absolutely delighted with that particular MOOC and the success it's had. And actually, people really like it. Okay, I'm getting near the end of it now, so big sigh here. But what I wanted to say is, I said it at the beginning, standing up in front of you, giving a talk like this, it's not a solitary thing, science. It involves an awful lot of people who you encounter along the way. And every single person that's on this slide has meant something to me. And whether they know it or not, they've really made a difference and impacted on what I did, and actually down to them that I'm here in front of you today. But I would especially like to draw attention to my family up in Scotland still. My daughters, who have been incredibly supportive and taken part in lots of the science communication stuff and have put up with an awful lot. And my husband as well, and I'm really sorry that you didn't go to Wembley yesterday to see Wolves lose in the semi-final, but I'm sure being here listening to me is much more interesting. And then you saw a picture of me graduating, but my daughter also graduated as well, so I just wanted to say that. Now, I wanted to finish by doing a short video. I had to think carefully about whether to show this or not, and I will give it a wee bit of a context. So Alan Partridge, he is a spoof character, okay? He's not real, but he's from Norwich, and this little video brings together antimicrobial resistance, equality and diversity issues, rabbits, and infection control. And I just thought it's going to work really, really well. He's very crass and he gets things wrong, okay? And what I've said here is that there's so much right with this and so much wrong. So you've listened to me tell you how I do communication, but watch how Alan Partridge does it. And I'm going to just finish with that because I don't think I can top it. But thank you very much.